Okay, good afternoon, everyone. We're going to go ahead and get started. Thank you all for coming to Medicine Grand Rounds. A few quick announcements. First and foremost, it is National APP Appreciation Week this week. The Department of Medicine sponsored their big event yesterday. I hope some of you had a chance to go. To the APPs in our audience and to those listening, thank you for everything you do. It's so nice to be able to appreciate you and have you be part of the faculty in the Department of Medicine. So I want to call out our APPs, and for those of you working with them today, take a minute to say thank you because they do incredible work. A quick reminder of our upcoming Grand Rounds. On October the 9th, Dr. Chifu will be here as the Meiklejohn Lectureship. He will be presenting on medical reversal, talking about cases and conundrums where he's had to make some difficult decisions, often going against the data, but also trusting his instincts. And it'll be an inspirational talk. On October 16th, Sarah Young will be here talking about a very interesting and important topic. What do the kidneys have to do with the climate? The intersection of change, kidney disease, and climate as well. And then October 23rd, one of our own from the past, Dr. Silk Soto, will be here talking about reclaiming the mission of academic health centers. Dr. Soto is at Indiana, has a leadership role there in the center, and is really thinking about the future of AMCs from a equity and inclusivity perspective. So I think it'll be a very inspiring talk. And to round out October 30th, Dr. Robert Califf will be here. Rob Califf is the commissioner of the Food and Drug Administration. He is in town really sort of doing a number of events with our local business community. I was able to twist his arm to join us here for Grand Round, so he'll be here. So please come. He's a tremendous speaker, and we get to hear about how devices and drugs are regulated in the US. Okay, with that background and some teasers, it is truly my privilege today to introduce my colleague, mentor, friend, and former boss. So I'm on my best behavior, Dr. John Carethers. I could spend an hour talking about John, but I'll give you the highlights in terms of his many accomplishments. He's a distinguished professor in the Department of Medicine, as well as the Vice Chancellor for Health Sciences for the School of Medicine at the University of California in San Diego. He also holds adjunct professorships at the Herbert Wythram School of Public Health and the Human Longevity Science, also at UCSD. John began his career with humble beginnings. He is a Detroit native. He went to medical school at Wayne State University, followed by internship and residency at Massachusetts General Hospital in Boston. Upon graduation, he completed his fellowship in gastroenterology at the University of Michigan. And following fellowship, he was recruited to the University of California, San Diego, where he stayed until 2009. In San Diego, he rose through the ranks to professor with tenure. He was immediately recognized for his leadership potential because everyone attracts themselves to John. He has this ability to have people come to him for all kinds of advice. So he became fellowship program director between 2000 and 2004, then section chief for the VA at San Diego Health Center, and then chief of the Division of Gastroenterology from 2004 to 2009. And this morning he shared with the chief residents how that happened. And he said no 12 times before he took that job, in case you're interested. John is the recipient of many awards. He was named the American Gastroenterological Association Distinguished Mentor in 2017. He's an elected fellow at the Royal College of Physicians in London as of 2021. He served as president of the AGA from 2022 to 2023. He's a member of the ACI, APM, and the National Academies of Medicine. John's career has really covered many areas, including leadership, but really has been consistent on the theme of colorectal cancer. He's been funded consistently since 1993 by government, private, and philanthropic entities, including the NIDDK, various NIH organizations, RWJ. He's authored more than 200 publications in high-impact journals. He's a recognized expert in this space nationally and internationally. It is truly a pleasure to welcome you here, John. Please join me in welcoming Dr. John Carethers to talk about environmental influences in colorectal cancer.
Thank you, Dr. Chopra, for that humbling introduction. And it's certainly a pleasure to visit this wonderful campus and meet many of your leaders and particularly the chief residents. I love meeting with the chief residents, so thank you. Let me see if I can get, I think I'm supposed to escape and switch here. Okay. I'm good. Turn that away because I have a lavalier microphone on. I'm going to talk a little bit about things that influence the development of colorectal cancer. And I'm going to do this in a way, when I say environment, I mean like the local environment, but also the microenvironment, but also the macro environment. These are my disclosures. They don't have anything to do with what I'm going to talk about today. And so I'm going to break up this talk into first talking about some environmental epidemiology, and then talking about how genetics can change the environment, and then how the environment can change the genetics of colorectal cancer, and tell you a little bit about many of the things that are going on in the microbiome environment. For those in GI in the audience, you know that for a human to develop colorectal cancer, it goes through some physical transitions as well as genetic transitions. I can switch to this if it's, because I don't want to get the feedback. Is there a way to turn that off? Okay. Hopefully that works. Physically, most cancers come from adenomatous polyps. And it takes a while for these polyps to develop over time. They require certain genetic characteristics, allows them to invade and metastasize. Most time we find benign polyps at endoscopy, but they can look a variety of different sizes and shapes. Some of these actually have cancer in them. If you think about what is really driven by the genetics and what's driven by the environment, we can look to what family history tells us. So if you look at approximately 150,000 colorectal cancers that happen in the United States, actually only about a third have any known familial component. In other words, either first degree or second degree relative that may have had a polyp or cancer. 2/3 is what we call sporadic, because there's no family history of that. The syndromes that you heard of, like FAP and Lynch, only account for a small piece of those. So what is driving this? And this is a very interesting study that was a mathematical model of about almost 50,000 pairs of twins. So they knew who was a monozygotic twin and who was a dizygotic twin. And they asked the question, what is the biggest driver? Is it the environment or is it genetics for 20 different cancers? Okay, and so as they analyzed this, it turns out only 3 cancers came out statistically significant for a genetic component. That was breast, prostate, and colorectal. Now, this is a mathematical model, so you have to take it with some grain of salt. But particularly for colorectal cancer, they said 35% what drove a colon cancer came from the genetics, but 65% came from either non-shared or shared environmental influences based on this mathematical model. So they concluded in this New England Journal of Medicine paper saying that the environment had the overall principal and dominant roles in causing sporadic cancer. We think of individuals. We have 8 billion people on this planet. We are all genetically unique. We look different. We have lots of different SNPs amongst us. And meaning if you add up all the potential genetic changes, you will have a slightly different risk for an environmental interaction than I will across those 8 billion people. And you can take something in environment, like a virus exposure or high-fat diet exposure, and your risk for developing a type of cancer is going to be slightly different. And I'll show you some examples of this. The way I think of colon cancer, and hopefully the way you think of this as well, is that we have modifiable and non-modifiable risk factors. And those 2 things add up to the ultimate risk for developing colon cancer. The non-modifiable are your genetic susceptibility. That could be if you carry a germline mutation like an FAP or Lynch syndrome. And there are high-risk alleles for that, and there are low-risk alleles for that. And you also, based on your ancestry or your genetic background, have a varied risk. And this is true from what we see even amongst the races and ethnicities in the United States. The other thing that's a non-modifiable factor is your age. Tell me if you can change your age, let me know. I will write a Nature paper together. The modifiable are generally the things that are in the environment, you know, susceptibility to your diet and hormone replacement therapy, the use of aspirin, those kind of things. And one thing that we as a medical group have control is the screening utilization rates, because screening is a part of preventing colon cancer. But those two things combined make the ultimate cancer risk. To put this to some thought here, if you look at the rates of colorectal, breast, and prostate cancer, they're all higher above the 37th parallel in the United States. And this is shown by the red versus the blue areas, and there's some spots in California. And some people have pegged this to the exposure to sunlight. And because sunlight, we take sunlight and we convert it to vitamin D, and vitamin D and calcium can lower your risk for colon cancer about 10 to 12% based on a lot of different studies. There are some hot spots in the United States that are below the 37th parallel, and this is along Appalachia and in the Mississippi Delta, where there is a lot of poverty. There are different background races from historically the way the country was set up with low education and don't have access to health care. And so when you look at the United States by breaking it by race and ethnicity as well, you're broken up by whites, blacks, Asian Pacific Islanders, Native American, American Indians, and Hispanics. The highest rates of colorectal cancer are amongst Native Americans. Those numbers surpassed already high rates of African Americans just 2 years ago. Okay, so Native Americans, you see overall incidence is 50 per 100,000 in Native Americans, now compared to 40.8 per 100,000 for African Americans. Okay, and the death rates have now surpassed. And there's a theory about this. Now that Native Americans are living longer, we're seeing the manifestation of cancers, particularly colorectal cancer, that we didn't see 20 or 30 years ago when many of them were dying in 30s and 40s and 50s. So for the last 30 or 40 years, it's been African Americans, the highest rate in the United States, but just in the last 2 years surpassed by now the longevity of Native Americans. The other interesting thing, and there's a lot more data on African Americans than Native Americans, is that even in early onset cancer, you can see here from one study, the rates of cancer were already higher in those under the age of 50, meaning they're preconditioned to getting those high rates compared to non-Hispanic whites or Asian and Pacific Islanders in this case. And in particularly for those from those more poor zip codes, the drive for some of this is based on the zip code you're born in. So if you're born in the wrong zip code, meaning that you're in a lower socioeconomic area, you're more likely to have a lower level of education, more likely to reside in a lower income, hold lower paying jobs, working several jobs to make things work, be in areas of grocery store deserts, et cetera, and have access to high caloric, high fat diet instead of healthy, farm fresh vegetables, and may be in even a medical care desert as well. And that has consequences down the line as you age. That can change based on what you're eating and what things you're not exposed to, or low physical activity can alter the gut microbiome and then have biological consequences of higher colonic proliferation. And then over time, that can increase your rate of colorectal cancer. And by the way, when you look at this is the same issue with when COVID first came out as well. So I can substitute colorectal cancer for COVID when COVID came out here. Why do we know this? It's because of studies like this. This is a study from Steve O'Keefe, who is at UPMC. And what he did, he took a group of rural Africans who are on a legume and rice diet. And then he took a group of African Americans from the South, I think it was North Carolina, and swapped their diets. He gave a high caloric westernized diet to the rural Africans who weren't used to that. And he gave the legume rice diet to the African Americans. And prior to the diet swap, he took biopsies of the colon before and after. And this was just a 2-week period. On the left, you can see that a proliferation marker, Ki67, for colonic cell proliferation was cut in half for the African Americans on the rural African diet and doubled for the rural Africans on the westernized diet. And if you look at the level of inflammation for intraepithelial lymphocytes or other macrophage markers, the level of inflammation dropped statistically for the African Americans on the rural African diet and significantly went up for the Africans on now the westernized diet. The other thing they looked at, it was some of the metabolic processes. Butyrate is a short-chain fatty acid. It is a great fuel for healthy colonocytes. And that butyrate is a good thing to have around. And it turns out the African Americans now on the rural African diet had increase in butyrate, whereas the rural Africans now on the westernized diet dropped their butyrate levels. And then if you look at bile acids, it's believed that some secondary bile acids are more carcinogenic, pro-carcinogenic, compared to primary bile acids. And so the African Americans on the rural African diet dropped their secondary bile acid production compared to the rural Africans on the westernized diet. So the bottom line is, just in a 2-week period, massive things changed in the colon. Can you imagine being on such a diet for 40 years or 50 years where you're having increased proliferation, different chances for genetic events to occur, and developing polyps and cancer? So that's the idea here. One of the other things, and you have Swati Patel, I think, in the audience here, is this conundrum of early onset colorectal cancer. This is another environmental thing that we don't fully understand. It is based on a birth cohort. Those born before 1960 never saw any increase in early onset colon cancer, those below age 50. But those born after 1960, those rates have been going up steadily every year, presumably based on some environmental exposure. So if you fast forward to the second, or this year diagnosis, so 28, 30 years later, the increases are all happening essentially under those age 50, where those over age 50, the rates are dropping, presumably due to screening. It's clearly this elbow goes up after 1960, and you can see the rates per 100,000 for the different 5-year bands for those under the age of 50. So why is there an increase in early onset colon cancer? Is it the same drivers that we see high for African Americans or Native Americans? They may not. African Americans have an excess of right-sided cancers, believe it or not, that are not microsatellite unstable. They are microsatellite stable. The cancers we're seeing early onset are left-sided. They're going up. So the zip code might determine something, but African Americans have more excess proximal versus the distal rectal ones. So are they the same thing? We don't know. It's clearly the most strong association has been with the environment and the diet, metabolic disease. The one thing that certainly not is screening utilization rates, like I showed in that previous table before, because we didn't screen anyone until 2 or 3 years ago for those under the age of 50. So what is going on? And these are hypotheses, excuse me, that there's some environmental trigger, excuse me, where you're exposed to something in the microenvironment, And that can happen at any time during the tumor initiation, progression, excuse me, or a carcinoma stage, and then accumulate epigenetic, excuse me, and genetic mutations that drive that early cancer. Is it the exact same as a late onset? And there's a number of investigations for this. This table, excuse me, sorry about that, taken from the TGCA data set published about a decade ago, kind of summarizes 30 years of genetic information. I have one right here, thank you. Sorry about that. And what we've learned on the genetic drivers of colon cancer. This upper table, excuse me, well, I'm coughing just right now. This upper table shows on the y-axis the mutation rates taken from the tumor. The x-axis has 4 areas: the site of the tumor and the microsatellite unstable status, and a marker, a target of one of the mismatch repair genes, MLH1. And you can see in this hypermutated group where there's tens to hundreds of mutations determined from the tumor, make about 15% of all colon cancers, and really concentrates the MSI status and the silencing of this mismatch repair gene. The other 85% of colon cancers only have a handful of mutations, 1 to 5, that drive its tumors, and the profile of those mutations are different for these different groups. So the hypermutated have a series of genes of which 2/3 have coding microsatellites. So you take out mismatch repair like MLH1, they have a coding microsatellite, they frame shift, you create a different protein, a neoantigen, and you can see the mutations are 1/4 to 1/2, so they're pretty consistent. The ones that have what I call follow the Vogelgram, where there's a handful of mutations, are classic. APC, P53, KRAS, TTN, and PI3 kinase account for the majority of mutations that drive those tumors. And then you have this long tail of mutations that are the fingerprint for every one of those cancers. But it's this 3 or 4 that really drive the tumors. And this has been shown now with organoid models where you can just take out APC and P53 and create a tumor. Whereas in the mismatch repair ones, that's not necessarily the case, although APC becomes secondary mutated because it does have coding microsatellites in that gene, and it's the gatekeeper for colonic neoplasia. How does the environment play a role in this? Well, this is data from the nursing health profession study. Andy Chan at Mass General has shown this data where they surveyed something like 150,000 nurses over 20 years. And this is secondary prevention of colon cancer. So these are people who had a colon cancer. And we know aspirin, like I talked about vitamin D and calcium, can reduce your risk for colon cancer by 40 to 50%. Not only for primary cancers, and there's some qualifications to that, but also for secondary cancer. So if you had a colon cancer, aspirin can reduce your risk. And what he did in this study is take people who they had a primary cancer, did a genetic fingerprint of those cancers, and find out the ones who had a mutant PI3 kinase or not. And you can see that the probability of death taken on the x-axis and the years since their diagnosis on the on the x-axis, that was on the y-axis, that if you had a mutant PI3 kinase, aspirin was greatly beneficial. But if you didn't, aspirin did nothing. So this is getting now to precision medicine and how aspirin can be you can select the right people to target for some of these things. And it turns out there's a biological reason for that because it downregulates wild type PI3 kinase. And so understanding that makes sense over time. So what drives can drive these cancers? So I showed that one graph for early onset. You have tumor initiation, progression, and carcinoma. We know clear-cut genetic lesions like APC for FAP and mismatch repair for Lynch syndrome. But sporadic, it's still, that process still happens. It's just the timing is different if you have a lead out of the gate because you had a genetic mutation. But anything in an environment, and I have listed here the microbiome and other aspects of inflammation, can influence any one of those steps and either accelerate or decelerate that process. I've had, and I'm sure Swati can test this too, had Lynch syndrome patients, same mutation, 3 generations in a family, and the oldest person, like the grandmother, will get a cancer at 50. The mother, the daughter, didn't get a colon cancer, and then the granddaughter gets a colon cancer at 25. Why is it different with the same mutation in the same family? Well, there's other, they're not quite genetically similar, even though they're in the same family, but also there's environmental things that can drive and influence that progression. The other thing I want to mention is the immune system. The immune system is such an important aspect. Even though I'm showing you a paper from 2006, this has changed oncology care. This paper was a landmark paper by Jeremy Gallone out of Europe, showing that he could predict the outcome of cancer patient better than any staging by just looking at whether you had immune cells in the center of the tumor or in the periphery of the tumor. And you can see the different stages, but he can group them. If you had CD3, which stains any type of CD cells, either at the center of the tumor in CT or the invasive margin, if they were high, meaning there was probably some protective piece to that, your survival was pretty good, irrespective of your stage. But if you had low, your survival was worse. And some have advocated that this immune staging is a better way to stage than classic staging of tumors, but it hasn't gotten fully into the mainstream yet. So let me get into where genetics can change the environment. And this has to do with DNA mismatch repair. So I'm just going to briefly go through this. DNA mismatch repair is an evolutionary conserved enzyme system that repairs DNA after DNA is synthesized in every cell of your body every day or every hour that you're proliferating in these cells. When you inherit a mutation or you inactivate one of these is when you can get either an inherited or sporadic form of microcell-like cancer. They work in, these proteins work in heterodimers, so MSH2 binds to 6, or MSH2 binds to MSH3, and then there's a secondary piece of this mismatch repair where MLH1 binds to PMS2. The bottom line is they recognize the newly synthesized and a template strand of DNA and lead to eventual exonuclease to carve out the mismatch that occurs, here shown by a CT mispair. And these are insertion deletion loops at microcellite sequences. And then the polymerase comes and resynthesizes the correct nucleotide or series of nucleotides for repair. So I call this the broken zipper model, shown here, where you might have a tetranucleotide sequence, and that any of these AAGs can bind to TTCs on the other strand. But if they don't quite pair up, you get either insertion loop, depending on if it's on the template or newly synthesized strand of DNA, because it could flip on the opposite side. And then that is recognized by these mismatch repair proteins, and then takes out the wrong number of these repeats, and then resynthesize hopefully with the correct number, okay? So that's how mismatch repair works in essence. Why is this important? Well, it turns out 15% of all sporadic cancers have this defect. They display microcell instability. 70% of these tumors are in the right side of colon. They're more common as we age, unless you have the germline form of it. They show poor differentiation. About 40% of these show mucinous features. So they look bad under a microscope, even though the outcome of the patient is actually much better than someone without it. The tumors are hypermutated because of all these nucleotide mismatches that happen. They're mostly diploid, so they don't have chromosome breakages and loss. But the key is that they attract T cells. Why? Because we have about 500 genes that have coding microsatellites, including APC, that frame shift and create neoantigens that attract your immune system because they're unrecognized normal human proteins. They are resistant to 5FU, and so I'm not going to go into all that. And again, these patients, when you compare it to someone who doesn't have this, actually have better survival. And so some of the key studies started over a decade ago when they were looking at tumor-infiltrating T cells in MSI tumors, and they could predict, based on these coding microsatellites, where the truncation of the new protein would be, because you know where the location of these, creating a stop codon and this neoantigen. And in people who have a microsatellite stable tumor, their immune cells did not recognize any of these. But if you take someone who had either a sporadic MSI-high cancer or a germline MSI-high cancer, a Lynch syndrome patient, they recognize this neoantigens, meaning their immune system is already, quote unquote, pre-vaccinated to these neoantigens. And this is one of the reasons why you see this intense immune response and lymphoid response to these MSI tumors for these patients. Even in a Lynch syndrome patient who has not had a tumor, they must be making small amounts of this because their immune cells recognize these neoantigens even without the presence of a tumor, meaning they're self-vaccinated against this. Okay? This is shown again by Jeremy Gallon's work later. This is a Muni paper a few years ago showing that if you have an MSI tumor, their memory T cells for mature T cells are way higher because of this recognition of these new stop codons and these neoantigens on those tumors. And the way I think of this is that you lose mismatch repair, whether it's a germline or sporadic, and then there's some genetic events, and about a quarter of these will have another mutation called BRAF, but now you're generating these neopeptides that are immunogenic from all these new stop codons that get recognized while you're creating more and more mutations. And because of this, they're producing these protective T cells that come into the tumor. It's like the prison guards surrounding the prisoners in jail as you're picking up the detection of microsatellite instability. And that protective immune response helps prevent until it escapes out of it, helps prevent the metastases of these tumors. So you have alteration of mismatch repair. You create these mutations, they induces this T cell protective, and the biological consequences of that environmental change induced by that genetic change is reduced metastases in these patients. This is borne out by one of the biggest revolutions in oncology over the last decade, and that's immune checkpoint therapy. This is the article that was in the New England Journal of Medicine showing if you have a mismatch repair deficient tumor, MSI-high tumor, that the change in tumor markers drop dramatically with immune checkpoint therapy. And you can see the radiologic, you see all the blue is they got better by just giving immune checkpoint therapy. And the performance-free survival, survival is much better with deficient mismatch repair or these MSI-high tumors. And that's been subsequently shown and led to the first ever FDA approval of these drugs for any tumor, whether it's colorectal or anywhere in your body, as long as they had deficient mismatch repair to use these drugs. It's the first time a non-organ-based tumor drug was approved for that. So the way I think of this is you can have germline or somatic inactivation. You get all these frame shifts and point mutations, even though most of these are non-coding, but there's about 500 of these that generate these immunogenic peptides that that can respond, the immune system can respond. And then the precision piece of this is improved survival and immune checkpoint susceptibility. So I'm gonna flip this. So how can the environment change the genetics in colon cancer? So just to recap on mismatch repair, I mentioned these proteins like MSH2 can pair, shown by these double-sided arrows, with MSH3 or MSH6, but then MLH1 pairs with PMS2. But the type of microsatellite generation is a little bit different depending on which of these proteins are taken out. We measure MSI high by mononucleotide or dinucleotide instability. That's why I have it here. There used to be this entity called MSI high low, which turns out was really MSH3 defect. And then there's another type of microsatellite instability we call EMAS, standing for elevated microsatellite alterations at selected tetranucleotide repeats. And that perfectly overlaps with this function of MSH3 as a sole takeout. Now you say, well, no one's told me about MSH3 before because we don't necessarily test for that in Lynch panels, because the reason why that you don't see is because there's some redundancy with MSH6, as you can see with this overlap, but probably those patients are 80 and 90 years old. But there's a biallelic form in which you do get a polyposis syndrome, but you have to have both of those taken out in your 30s. So what are some common features of EMAS? And one of the reasons I started studying this a few years ago was because we saw this EMAS in 50% of all colon cancers. But we didn't understand it at the initial thing. So that's, they display these EMAS, so this tetranucleotide instability. The cancers aren't specific to their proximal like classic MSI high tumors, and they're not hypermutated when you're looking at MSH3 inactivation, and that's because most of the coding microsatellites are mononucleotide repeats, and when you take out MSH3, you have no frame shifts of mononucleotides. Let me go back here. You take out MSH3, there's no problem with repairing mononucleotides where most of the coding microsatellites are. The other thing, they're sensitive 5'U". We've done that study before. Unlike classic MSI tumors, they have shorter survival and poor prognosis. And we've published they have higher frequency among African-American tumors, suggesting that this may be one mechanism that may be leading to that higher poor survival rate. So this is a study from my former mentor, Rick Bowen, that was published in 2012 when this was starting to be discovered. And you can see in this cohort of patients that they had, those have MSI had great survival, and they get immune checkpoint therapy and they get even better survival. But if you had this EMAS form, you didn't have such great survival, and it was statistically significant like stage. But no one knew the driver or cause of this. There were several papers eventually shown, and we were one of those groups, that this is driven by a defect in MSH3. Now, why do you get an MSH3 defect? We looked at germline mutations, somatic mutations. So even though we kind of knew MSH3 was the target, we couldn't find an etiology. We looked at methylation and everything. So what was causing the MSH3 defect when you can't find a genetic effect that's driving its inactivation? Well, one of my postdocs did an accidental finding, and that's how discovery happens. And she was doing some immunofluorescence. We were putzing around with an MSH3 antibody, and she happened to notice that some of the staining in the nucleus seemed to be off. So then she tried to chase down some studies, and this was one of the first studies where we took a cohort of EMAS positive tumors, and we were looking at pro-inflammatory cytokines because there was some relation with inflammation. And we showed this correlation of EMAS positive being more associated with more expression of IL-6. And this was the key experiment that we published about 9 years ago, where we used the combination of immunofluorescence and Western blotting showing that if you take an antibody, gets MSH3, okay, here's control. You see all the greens staining these nuclei, and then DAPI stains the nuclei, and you see the overlap of all the MSH3. A DNA repair protein is where the DNA is in the nucleus. But if you give IL-6, the MSH3 disperses out of the nucleus where it can't repair the DNA. And then this is seen here for MSH3, where we give increasing dose. We separate the nucleus from the cytosol by kind of like separating the egg yolk from the egg white of the cells. And as we give increasing amounts of IL-6, you see the amount in the nucleus drops dramatically, 100-fold, while picking up in the cytoplasm more than 100-fold. And this did not happen with the other mismatch repair proteins. So this strongly suggests that inflammation was driving the change in MSH6. So it was no mutation, it was just shifting out of nucleus where it couldn't repair. And we showed that here by taking an intrinsic tetranucleotide sequence, MYCL1, and showing that after a week or two of IL-6, we had 8 to 10-fold more mutation or frame shifts of that intrinsic tetranucleotide sequence with just IL-6 treatment, suggesting that this movement really inactivated the MSH6. So and without going to a lot of other details, so I'm really truncating this whole story, but the story is that the inflammation and neoplasia might start, but then you have, the initiation of neoplasia may start, but inflammation occurs, and that's IL-driven. And by way, we looked at other pro-inflammatory cytokines. It's IL-6. And like I said, I'm skipping a lot of pieces of the story. Then there's a loss of function of MSH3 because it's not in its right location. And then that may have potential di, tri, and tetranucleotide frame shifts because MSH3 is not functioning, but not mono. But the other thing I didn't tell you about is, and this is a number of series of papers as well, that MSH3 is also involved in homologous recombination in the BRCA complex, and it may induce double-strand breaks. So I'm skipping a lot of this story, but the bottom line is the combination of this is different than classic MSI in the biological consequences is increased metastasis and poor outcome. So putting these two together is, the up top I went over for the classic MSI, but on the bottom, the inflammation takes out MSH3. You get these frame shifts at these longer microsatellites, but not the mononucleotide ones. And you get DNA double-strand breaks that leads to the poor survival. And we're still studying this aspect, and I can talk a little more about that. But I want to tell you some other findings about the environment that are very exciting and are going to have implications on how we diagnose, screen, and eventually perhaps treat colon cancer. So I showed you this, and I imply here that inflammation microbiome as part of the environment can influence any aspect of the pathway for developing colon cancer. Now, there's been a lot of thought over the last couple of decades on, looking at the microbiome in the colon. The colon is probably the most microbiome dense region in your body. There are something like 10 to 6 to 10 to 9 bacteria. A lot of them are, pass through, a lot of them are resident, but they influence things. About bacteria that can process, your secondary, from your primary bile acids, about bacteria that can help you produce certain vitamins, et cetera, especially some that are in the small intestine, et cetera. But bacteria and influence either initiating or influencing colonic neoplasia has been an idea that's been around. The question has always been, what's the chicken and what's the egg? So, we could have normal epithelium. Is the proliferation because of some genetic event, or is it some induction from the bacteria that happen to be in your colon at the time? And I showed you from O'Keefe's study, by diet swaps, you can change a lot of these things, including the bacteria, in a 2-week period. okay? And then as the tumor potentially grows, there's association with microbiome on the surface. So again, is that polluting or synergizing with the growth, or is it just a bystander effect? And so this has been a question that's been around, but there's been more serious details now over the last 5 or 10 years that have come to this. There's been some studies looking at associations. So this shows, and I'm going to talk about these three briefly, about particularly colon-producing E. coli, which we all have in our gut, enterotoxigenic B. frag, which we also have in our gut, and Fusobacterium, which lives in our mouth, but is associated with colon cancer in your gut. And all three of these things have been associated epidemiologically with microbiome enrichment, maybe not strongest with immune responses, but they also make toxins that may influence things too. Colon-bacterium producing or PKS positive E. coli and the BFT toxin B. frag as examples. This is a fantastic study that just came out about 5 years ago from Cynthia Sears' work at Johns Hopkins University, where she took FAP patients, which we know makes lots of these polyps before they get the cancer, and controls which didn't have FAP and polyps. And she looked specifically at the PKS positive or colon-bacterium producing E. coli, enterotoxic G. frag, and then a combination of two. And you can see in FAP patients, they were more likely to have both of those associated with their polyps on microfilms compared to controls, okay? And then if you flip it around with neither was more high in the controls where they were less likely to be neither in the controls. So that was human patients. Then she said, okay, I'm gonna take a mouse model and give them these bacteria and see what happens. So she took the FAP model, which is an APC mouse model. So they have a germline mutation in this gene that gives them lots of polyps. And so she gives them the B. frag, the E. coli, and the combination. So with one or the other, the mice lived about 110 days, but when she gave them both, they succumbed more like 80 days. And then she looked at what happened in the colon and along these polyps. So here's sham, not much inflammation score. Either the B. frag or the E. coli, not much difference, but the combination two seemed to rise it. And she also took a mouse that couldn't make the pro-inflammatory cytokines. And when you take, in this case, an IL-17 knockout mice, they don't make the tumors, suggesting that these bacteria are doing something to influence the inflammatory pathways. Like I said, the pro-inflammatory IL-6 in my study. So these combination of the two certainly increase inflammation, have faster tumor onset, poorer survival, and were definitely pro-inflammatory cytokine dependent, okay? She has also done some other work with other bacteria. This is a study from human colon tumors, and you can see this concentration of red ones where she looked at the presence and acquiring of Fusobacteria. I said Fusobacterium nucleatum is an oral bacteria. So why are you picking this up in the colon? And you can see this aqua color is also picked up where there's biofilms in people who have tumors, but you really don't see it in people who have biofilms without the tumor. And this is found in the majority of these right-sided tumors. Of course, we think of right-sided tumors as MSI-high tumors, of course. And it turns out the risk of proximal colorectal cancer was 5-fold higher if you had a biofilm containing this Fusobacterium. And so the supposition is that this association might be associated with some of these right-sided type of genetic pathways like MSI-high tumors. But keep in mind, this is oral. So is it getting down there by somehow surviving through the entire 20 feet of intestine and residing there? Or do you get some type of bacteremia and it reloads there? It's an oral anaerobic bacteria. So it lives in some of the anaerobic areas of the mouth, and presumably the most anaerobic area of the colon is the right side of the colon, and maybe it thrives there. Okay, so also in her study, she looked at okay, is this driven by inflammation? And certainly the ones at the biofilm, they had higher levels of IL-6, and that was and then one of the downstream signalings is the STAT3, so those were both higher. And even the intraepithelial cells had higher levels of IL-6. And if they're biofilm positive, the proliferation rates of even the normal crypts around them, you can see much more brown staining showing proliferation, Ki67 staining, were even positive on the normal crypts around them, suggesting that the presence of these biofilms was inducing colonic proliferation and maybe synergizing with the development of these polyps and tumors. This is a study we published 3 or 4 years ago, just looking at, there was no study out there suggesting as Fusobacterium toxic to any colonic cells. And what we did is we took Fusobacterium, cultured them with cells, then took the supernate and put it on cells and see anything it made, did it do anything? And turns out it caused DNA damage. That was the first study out there that was published. And this is tested by the presence of double strand breaks stained by gamma H2AX. And you can also see if we gave metronidazole, which is an anaerobic anaerobic bacteria-affecting antibiotic, that we can prevent the double strand breaks by killing the bacteria first, and then whatever it was making didn't cause the DNA damage. This is also a study we published about not quite a year ago, where we looked at over 300 colorectal cancer cases, of which about a third had presence of Fusobacterium nucleatum. And you can see the breakdown of about almost half were microsat stable. 42%, a little bit less, had this EMAS phenotype. Remember I said it was about 50%. And about 12.5% had MSI high. And we showed the strongest association of Fusobacterium with the MSI high, but it was also associated with the EMAS, but to a lesser extent compared to MSS, suggesting it might be driving some of these processes. We also looked at odds ratio showing some connection with MLH1 hypermethylation. That means it's tied to MSI high. BRAF mutation, again at MSI high. Even KRAS mutation to a lesser extent. So multivariate showed these 4 things, and KRAS was the oddball, but when we did multivariate analysis, we found, and without spending too much time on data, that is associated with these, the KRAS seemed biologically different, but on the multi-analysis, and it was not a prognostic indicator for survival. But then we asked the question, so what is driving this? So what's common to these? These are all pieces of the serrated cancer pathway. Every one of these, BRAF, progressive methylation of target genes, targeting MLH1. So we believe that the Fusobacterium, based on that analysis of 300 cancers, is getting these tumors based on the serrated adenoma pathway and not by any other pathway. The other thing, and just to close this out, and I know it's 12:50, this is data from Susan Bullman and Matt Meyerson. It was published again a few years ago, which was just unbelievable data. But I want to point it out to you. So they looked at Fusobacterium in the primary tumor. These are 2 examples of primary colon cancer. They looked at a number of these, low-powered and high-powered, and the person's paired metastases in the liver. So this person was stage 4, and they stained for Fusobacterium presence. And you can see that there's Fusobacterium in this kind of microfilm, and it's associated with this, collection of tumorigenic cells in the primary colon. And when they stained the metastasis, they would find the Fusobacterium in the liver metastases. And shown by these this is RNA in situ hybridization, okay? So the question is, okay, so you got this Fusobacterium in particularly these right-sided tumors. How does the Fusobacterium get to the liver? Does it metastasize with the tumor, meaning it follows it, or does it secondary colonize the tumor? And what they did is they did a serial passage through mice where they took a human xenograph of a colon tumor that had Fusobacterium in it, several of these, and they did serial passages through mice, figuring out that it would peter out. But what they found was that they were able to pick up the Fusobacterium by culture by 4 or 5 passages of mice, suggesting it stuck with the tumor, and they were picking up on 8 serial passages by PCR. Their thinking is, this goes with the tumor. It's not a secondary colonization. So when we think of metastasis, we think, oh, the tumors acquired all these ability to get through vasculature or lymphatics and reside in a new thing. But this suggests it's carrying bacteria with it. Okay, that's a novel concept. So they did this, they took a regular tumor line that didn't have Fusobacterium, and then they took one of the human xenografts that had the Fusobacterium with it, and they treated the mouse with metronidazole. They shrunk the tumor by 30%, meaning an antibiotic killed whatever the bacteria was synergistically doing and prevented the accelerated growth of that tumor to 30%. So that tells me there's synergy between the Fusobacterium and the cancer cell itself. So just to close out, we have now 3 understood genetic pathways, what I call chromosomal or instability, or the Vogelgram model, which is 85%, the 15% for the MSI high, where there's a germline lynch, and 20%, which we call CPG island methylator phenotype, which gets to some of these serrated pathway, and this overlap between these. And the pathways are slightly different, but you get colon cancer at the end. But they can be influenced by this EMAS, which can influence all 3 of these pathways with inflammation. And just to close, the macro and microenvironment plays a major role in cancer formation, progression, metastases. Colon cancer is a great example. This is probably true in breast cancer, prostate cancer, and some others. Understanding the role can help us guide. I showed you one of the precisions with aspirin and PI3 kinase. There's other things that are coming down the line that's going to be microbiome based and others in addition to screening utilization. So I'll stop there. I know it's 12:56, I think. Okay.
Dr. Carethers, thank you. What a great talk. So many questions, so little time. I guess maybe I'll get started. And, the IL-6 story is very interesting because I was thinking about IL-6 inhibitory drugs and the potential role in colorectal cancer. Do we have data in studying this in humans?
So using something like tocilizumab will probably be an expensive prospect to do that, because that level of inflammation that we saw with EMAS is in half of all colon cancers. It might be easy to use things like aspirin and other things that might be able to reduce inflammation and way, 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 way cheaper. Of course, there's side effects to aspirin, of course. And we do know from, I'll say, some of the genetic trials, like with Lynch syndrome, the CAP trial, the CAP2 trial, that aspirin can even reduce the onset of cancer to some degree in even genetic germline patients like Lynch patients. So no one's ever done a trial of it, like with one of these anti-IL-6 drugs or anti-IL-17 or any of these blockage of pro-inflammatory. I think it's an expensive prospect, and I think that inflammation is probably driven by the diet and the other aspects of the environment that help create this environment and cause the secondary effects of loss of MSH3. So I think we can probably do it with that. The hard part is it's hard to get people to change their lifestyle. Everyone wants a drug or a pill. Like, I've been eating like this for 40 years. And some people, it's the product of their macro environment, as well as I talked about the zip code you're born in. So it's easier to do it by screening. And we can reduce and eliminate disparities with screening. We can show that. It's much easier to do that if we have access to screening for everyone versus doing a lifestyle or using an expensive drug. So no one's done a trial with tocilizumab, but I would probably one of these other aspects before we throw a $1,000 or $10,000 drug on something like that.
There's a question back here.
Thanks, John. Excellent presentation. Swati Patel, RGI division here. The Fusobacterium story is really interesting, and a lot of the data that you showed were from colonic samples.
Yes.
My question is it reliably shed in the stool sufficient enough that it can be a part of stool-based screening programs in a lot of these multi-omic type screening tests that we're starting to see?
Yes, so I am aware of, in the pipeline, but not ready for clinical use of people using proteomic microbiome data for screening for neoplasia. So you know that we have DNA screening, now we have RNA screening, fecal tests, now we have some of these blood tests that aren't quite as sensitive as the fecal tests, but there are tests coming down the line that are in various aspects of the pipeline that's going to involve some of these, including Fusobacterium. And Fusobacterium is a great one because the PKS positive E. coli and the enterotoxic B. frag, we all have that. And you might have a polyp, and a little polyp, okay? No one's going to die over a little polyp, or maybe even a modest sized polyp. And so there's going to probably be a lot of false alarms. But Fusobacterium is less associated with polyps and more with cancers. There's some on the polyps, but more associated with cancers, and that's probably going to get a bigger bang for your buck if you're focusing on colon cancer. I also want to get the advanced polyps, as I'm sure you do as well. And there is some advanced polyps that have Fusobacterium as well. So yes, some of these are in evolution in various labs around the country and perhaps around the world. They're just not for prime time. And I think, in my guess, in the future, as these different screening modalities to try to get 80, 90, 100% of the population screened, it's probably going to be a mixture of some of these modalities where we can't do colonoscopies on everyone. So some of these things that might mix a genetic piece with one of these proteomic or microbiome piece might be getting those sensitivity specificities up for the things that we want to, the advanced polyps and, what the DNA and the RNA are around 43, 45% for advanced polyps, and they're 90 plus 100% for cancers. Can we get the polyps up to 60 or 70 with some of this mixture of stuff, and time will tell.
I guess one last quick question. I'm Blair Finnemore. We met just briefly before you started. I do IBD here. We know that IBD patients have a higher risk of colon cancer, and that's really driven by inflammation.
Yes.
And the consistent theme in your presentation was inflammation by any variety of pathways drives cancer. I guess kind of getting back to what Swati was saying, is there any of these processes that or assays that are coming out that are looking at inflammatory markers, be it on biopsy at the time of screening colonoscopy or in the stool, that might inform somebody's future risk beyond just you grew polyps and so you're at higher risk?
So your question about inflammatory markers and risk are apropos. They're just not as specific if you do it in the blood, maybe in the fecal. So there's been cardiovascular risk, higher inflammatory markers, your risk is higher, right? They use CRP and other things. In the colon you can think of it as, can I detect early neoplasia? Can I detect neoplasia itself? And can I prognosticate it? That's probably the three things you're trying to think of. For detection, just doing a blood draw for a pro-inflammatory is probably not going to be very specific, because we could have any inflammatory process going on in our body at any one time. You bruised your knee, you're whatever, and it could go up, or cardiovascular risk, or whatever. So it's not going to be very specific. But once you have a cancer, could it prognosticate or a recurrence? The real question is the specificity, because it is up in people who have cancer and some advanced polyps, but it's up in a lot of other things. And so the specificity is the key thing.
John, thank you very much for a wonderful talk.
My pleasure.